used to, now if you take our genes, this can be seen. Okay, in what shape the histone proteins are arranged?
Cubic shape.
Cubic shape. Okay, and DNA is coiling around it. This is what we call histone associated with DNA. So this arrangement can we notice in the domain bacterial DNA?
Yes, sir.
No. In archaea, what is given to you? In archaea, in the table, what is given to you regarding this?
Present in some genes.
Some genes, correct? Present in some genes. Okay, some species or some genes, which is right?
Species.
Some species, not in some genes, present in some
Species.
species. What about eukarya?
Present in many species.
Written that way? Is it written that way? Present in many species?
That is present in many genes.
Present, another, correctly reading, no? So, in all the eukaryotes, there is histone associated with DNA. Understood? Hello?
Yes, sir.
In all the eukaryotes, DNA is associated with histone, or histone is associated with DNA. What about archaea? All the archaea has histone associated with DNA. Is it correct?
No, sir. No, sir.
No. None of the domain bacteria have histone associated with DNA. What do you say?
Correct, sir. Correct, sir.
Correct. Correct. All the prokaryotes, okay, all the prokaryotes, okay, don't have histone associated with DNA. What do you
Wrong,
say?
sir. Wrong,
Wrong.
sir.
Wrong. Correct. Should be careful in those kind of statements. Okay. Archaea is a prokaryote, but has, in some species at least, histone is associated with
DNA.
DNA. Clear?
Yes, sir.
Right. Next it is, what kind of chromosomes?
Circular chromosome.
What kind of chromosomes?
Circular.
Circular chromosomes. Okay. If you take a chromosome, that's an important thing to tell you, right? Chromosome have DNA, but all the DNA are not coming under chromosome. Understood?
Yes, sir.
Understood?
Can you repeat that again, sir?
If you take the DNA in a cell, okay, in that case, now if you take the DNA, now let's take like this, DNA in a bacterial cell. Okay, if you take DNA in a bacterial, I'm taking first for the bacterial cell. Okay, here in the center of the cell, there will be a DNA called what name you call it?
Central DNA.
Central DNA. In addition, there will be circular DNA molecules called, what is it?
Plasmid DNA.
Plasmid DNA.
Plasmid DNA. This central DNA is a what kind of DNA?
Circular DNA.
Circular DNA.
Circular DNA. This plasmid DNA is also
Circular DNA.
Circular DNA.
circular DNA. In these two, for the central DNA only, the name given by the word, what is it?
Chromosome.
Chromosome. So this is also DNA. That is also DNA, correct?
Yes, sir.
Plasmid is also there. It is also DNA, but it is not called by the name, what is it?
Chromosome.
Chromosome. The reason in the DNA, later in the unit number 7, you will study that one in full detail. Okay, it has a place called Ori. What is the name of the place?
Ori.
Ori. We will later discuss what is Ori and what it does. That is not our issue. So here, what is it?
No.
No.
If because Ori is absent, we can't call plasmid DNA by the name, what is it?
Chromosome.
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 Are you clear?
Yes, sir.
Yes, sir.
Right. That means what I told you in the beginning is all the chromosomes have DNA. Correct?
Yes, sir.
Yes, sir.
But all the DNA are not chromosomes. Agree with it?
Yes, sir.
Yes, sir.
Right. So take this one down. It is your syllabus. No problem. You can take it up.
Done, sir.
Done, sir.
Okay. Similarly, if you take a plant cell, or you can take a eukaryotic, what is it? Cell. I am taking a plant cell, DNA in which cell?
Eukaryotic.
Eukaryotic cell. Here there are we can divide DNA in nucleus. DNA in, what is it?
Nucleus.
Nucleus. This DNA is what kind of DNA?
Linear.
Linear DNA.
Linear DNA. It is getting the name called, what is it?
Chromosome.
Chromosome.
Chromosome. Understood?
Yes, sir.
On the other hand, in the eukaryotic cell, there are mitochondrial DNA. It is a circular, what is it?
DNA.
DNA, and there is chloroplast DNA. It is also what kind of DNA?
Circular DNA.
Circular DNA. They are not chromosomes. They are not, what is it?
Chromosome.
Chromosome. So we don't say mitochondrial chromosome then. Understood?
Yes, sir.
We don't say chloroplast chromosome then. They are in the stage called DNA. Understood?
Yes, sir.
Yes, sir.
So, would you please take it down?
Done, sir.
Right. So, if you consider chromosomes in cells, you know all the DNA are not, what is it?
Chromosomes.
Chromosomes. The chromosomes in cells can be divided into two. One, we can say, what kind of chromosomes?
Linear
Linear
chromosome.
chromosomes.
Linear chromosomes. So linear chromosomes, okay, belongs to domain eukarya. Clear?
Yes, sir.
Linear chromosomes belongs to, in the same domain eukarya, there are circular DNA inside mitochondria and chloroplast. Will you consider that part as a chromosome?
No, sir.
No, sir.
Linear chromosomes are there in the domain eukarya. If you take the circular chromosomes, you know the meaning for now chromosomes, that can be seen in domain bacteria and domain archaea. So this is what's the meaning for that table comparison. They say circular chromosomes present in the
Domain bacteria and archaea.
domain bacteria, present in the
Domain.
domain archaea, absent in the
Domain eukarya.
Domain eukarya. Domain eukarya has circular DNA molecules. No, any student should argue, sir, circular DNA is there, no? Then circular chromosome is there, no? Mean they are not chromosomes. They are not in the category called, what is it?
Chromosome.
Chromosome. That's the point. You can take it down.
Done, sir.
So, are you clear with that comparison?
Yes, sir.
Yes.
Right. Thereafter, they are telling about another comparison. What is it? Introns in, what is it?
Genes.
Gene. So, I have to explain you what is meant by, what is it?
Introns in
Intron.
Okay. If you take a gene, here I am taking a gene. Say this is this is a piece of DNA which is working as a, what is it? Gene. In it, there will be two kind of regions. One kind of region called exons. Another kind of region called, what is it? Intron. Again, there will be, what is it?
exons.
Exons. Again, there will be
Intron.
intron. So alternatively, normally, now if you take a human gene, you can see this one. You can see in a human gene, these two sections alternatively occurring. What are the two sections?
Exons and introns.
Exons and
Intron.
introns. So this is Buddha, this is a which molecule?
DNA molecule.
DNA molecule. It's a gene. Okay, it's a DNA molecule mean it is a, what is it?
Gene.
A gene.
Gene. Okay. Now, in previous classes when we studied RNA, DNA, I would have told you from the DNA, it will be formed an mRNA molecule. What will be formed?
mRNA molecule.
An mRNA molecule will be formed. So when this is copied on the, what is it? mRNA, in the mRNA also, the exon followed by
Intron.
intron, then again
Extron.
exon, then again
Intron.
intron, then again
Extron.
Exon.
exon, and finally again intron. Okay, will be there. This is called initial mRNA. What kind of mRNA?
Initial mRNA.
Initial mRNA. Okay. Finally, now we are going to work on which mRNA?
Final mRNA.
Final mRNA. In the final mRNA, the introns, okay, you know, this is home
Exon.
exon, this is home
Intron.
intron. All the introns, now this is a this is an exon, correct? This is another exon. This is another, what is it?
Exon.
Exon. These exons will be taken, joined together, understood? And it will form the which mRNA?
Final mRNA.
Final mRNA.
Using the final mRNA only, it will be whom it is produced.
Protein.
The protein produced. The protein will have a amino acid sequence according to the whom final mRNA. So in the protein making, is there a contribution to introns?
No.
No, sir.
No. Only exons join together and make the final mRNA. That final mRNA is the one converted to protein. Due to this reason, okay, introns are known as nonsense, okay, codes. What is the meaning?
Nonsense.
Codes with no sense. Understood?
Yes, sir.
Codes with, what is it?
No sense.
No sense. Okay, actually, introns have some other functions. Only one thing, they are not contributing to
Forming a
Protein
protein.
synthesis.
the protein production. Okay, nonsense. You can say nonsense nucleotide sequence. Telling the code is a simple word. Okay. Why it is called nonsense nucleotide sequence? Because okay, it is not expressed in the final. Okay, it's not expressed, that's much enough. Or you can say it is not in the final, what is it? mRNA. So your table is telling whether this intron is present or absent in the genes. Understood?
Yes, sir.
Are you clear, everyone?
Yes,
Yes, sir.
Some of them have introns, some of them don't have, what is it?
Introns.
Introns. So could you please take it down? This is your syllabus. In the unit number 7, you will be fully studying it. At the moment, for this particular idea, to understand, without understanding, I can just tell and go. There are unit pieces called introns in the gene. Introns mean introns. That way also we can tell. Understood? Okay.
Excuse me, sir.
Yeah, Buddha.
Sir, can you explain this again?
If you take a DNA, in a DNA or a gene, okay, a gene, first you have to understand a gene is the one first finally converted to protein. Do you understand it?
Yes, sir.
A gene will form an mRNA. That mRNA will form the protein. That is how protein is synthesized. Understood?
Yes, sir.
If you take a gene, in it, two types of regions can be noticed. What are the two types of regions? What are the two types of regions?
Exon and intron.
Exons and introns. Okay. When an mRNA is made, at the first, exons and introns are copied to the mRNA. So the mRNA also will have home and home,
Exon and intron.
exon and intron. Finally, all the introns are cut removed using enzymes. After removing the introns, who are remaining?
Exons.
Exons. All the exons are joined. And that chain coming by joining all the exons is called by the name, what is it? Final mRNA. Okay. Protein is synthesized using the nitrogen base sequence in the final mRNA. That is clear?
Yes, sir.
Okay, nitrogen base sequence in the final mRNA is the one that decides the amino acid sequence in the protein. So, intron sequences, are they coming in the protein? Tell me.
No.
No, intron sequences are cut removed, correct?
Yeah.
So, intron sequences are not coming in the final protein. So, due to that reason, new introns are called by the name nonsense nucleotide sequence. Understood?
Yes, sir.
Right? It doesn't mean they don't have any function. They have functions, but they are not coming in the protein. Understood?
Yes, sir.
Done, sir.
Have everyone completed it?
Yes, sir.
Right. So, if you have understood what is intron, now understanding this is easy. Is there introns in the genes? In the domain bacteria, they say having introns are very rare, but they don't tell absent, correct?
Yes, sir.
In the domain archaea, they say their introns are present in some genes. In the domain eukarya, they say they are present in many genes. Even in the domain eukarya, we are not mentioned introns are present in all the genes. Correct?
Yes, sir.
Only the probability wise, more intron opportunities are there for the eukarya gene. Very, very low chances are there for
Bacterial.
Bacteria, but none of the domain is 100% missing the
Introns.
Introns in genes.
introns. Are you clear?
Yes, sir.
Yes, sir.
Right. So that is this genetic composition idea. Hope you understood it. Understood it?
Yes, sir.
Yes, sir.
Okay. Now we are coming to the table for further development. What is the heading?
Protein synthesis.
Protein synthesis. How many points are there for protein synthesis?
2.
2 points. One, it is RNA polymerase. Other one, initiate amino acids for protein synthesis. Okay. Could you please complete this one? Protein synthesis part.
Done, sir.
Okay. Have you all got it?
Yes, sir.
Right. So, if you take RNA polymerase is an enzyme. RNA polymerase is an
Enzyme.
enzyme. It is mainly taking part in making RNA from DNA. Understood?
Yes, sir.
What is the role of RNA polymerase?
Help
RNA polymerase. Okay, you have to understand this one. Okay, a DNA, okay, can be make copies of DNA. From a DNA, you can make, what is it?
Copies of DNA.
DNA copies. Keeping one DNA, several copies can make. Okay, that ability is there for whom?
DNA.
DNA. This nature is called by the name
Self-replication.
DNA replication. What is the name given?
DNA replication.
DNA replication. In doing this one, though there are many enzymes involved, many enzymes there, inshallah, in future, under unit number 7, you will study them in full detail. Just for an idea, there is an enzyme called, what is it?
Polymerase.
DNA polymerase. It is not the only enzyme. There are many enzymes needed, but mainly who is needed?
DNA
DNA.
polymerase.
DNA polymerase is needed. Similarly, if you take from DNA, RNA copies can be produced. Understood?
Yes, sir.
Okay, from DNA, who can be made?
RNA.
RNA can be made. This is called by the name transcription. What is the name given?
Transcription.
Transcription. This is the only way to produce RNA. The only way to produce RNA in a cell is it should be made from whom?
DNA.
DNA.
DNA. Okay. And the enzyme involved here is called
RNA polymerase.
RNA polymerase. Main enzyme only I mark. Okay, this is not unit number 7, no. Just for an idea, I am marking. Okay. Similarly, from RNA, this is in some bacteria or virus only. From RNA, who can be produced?
DNA.
DNA can be produced. This is called by the name reverse transcription.
DNA.
What is the name given for this?
Reverse transcription.
Reverse transcription. Okay, transcription is DNA to RNA, no? Correct?
Yes, sir.
Yes.
Reverse transcription is RNA to DNA. It is
DNA.
done by
DNA.
an enzyme called reverse transcriptase. Understood?
Yes, sir.
Yes,
Yes,
sir.
sir.
But remember, there's no any way to produce RNA from
RNA.
RNA. This conversion, okay, we can say not possible. Not possible. If it is making, it will make like this. If it is making, now RNA is there, you need RNA copies, mean first from the RNA, which one will be made?
DNA.
DNA will be made. From the DNA, now RNA will be made.
RNA.
In that indirect way only it can be formed. Okay, first it has to perform a which kind of process?
Reverse
Reverse
transcription.
Reverse
transcription.
transcription.
After completing reverse transcription, there will be a DNA copy in your hands, correct?
Yes,
Yes,
sir.
sir.
Yes. From the DNA copy, we can, the organism will complete the
Transcription.
transcription process. Understood?
Yes, sir.
Yes, sir.
Right. So RNA polymerase is an enzyme related to making RNA from
DNA.
DNA.
DNA. In this one, okay, one kind of RNA polymerase there, or more than one kind of RNA polymerase there, is the comparison. Understood?
Yes, sir.
Right. So anyway, this is your syllabus. As I told you, certain points, what is coming in the future related to my discussion I am giving here, and that will be a good thing to you in future when you learn those lessons somewhere in your memory in a corner. Okay, sir told like this. That will come to your mind. Understood? So it's not wrong. But one thing, DNA polymerase, reverse transcriptase, they are not in the unit number 3. Okay, don't blame me or your school teacher when she is teaching. If she doesn't mention, that doesn't mean that I have taught you more. Understood?
Yes, sir.
Okay, it is not related to unit number 3, but it is related to your syllabus. So just I can just RNA polymerase just touching and going is not worth. Have you completed it?
No, sir. Done, sir.
Okay. Are you
Yes.
clear regarding RNA polymerase?
Yes, sir.
Right. Another thing it is, what is the next property? What is the next feature? Initiator, correct?
Yes.
Initiator amino acid in protein synthesis. Remember, during protein synthesis, amino acids are joined one by one, no? Correct?
Yes, sir.
During protein synthesis, one amino acid is brought, another amino acid is connected, then another amino acid is brought, that is connected, another amino acid is brought, that is connected. That is how, what is it? The protein synthesis takes place. Okay, it is called by the name translation. Now look at this diagram here. Okay, in the beginning, this amino acid came. Thereafter, this amino acid came. Then between the two, peptide bond was formed. Understood? Thereafter, this amino acid came through tRNA. Then for those two, this is also joined by peptide bond. Like this only, one by one, all these amino acids are getting joined. Understood?
Yes, sir.
Right. In this one, the first joining amino acid is there now. The first joining one. The first joining one. The first joining one is the initiator amino acid. Clear?
Yes, sir.
Yes, sir.
Right. Accordingly, in this diagram, what is the initiator amino acid? Glycine. GLY, they have written. GLY stands for, what is it?
Glycine.
Glycine. But remember, a protein synthesis will never start in glycine. A protein synthesis will start in the amino acid called methionine. What is the amino acid?
Methionine.
Methionine. Okay. Methionine is the starting amino acid in most of the time. But in domain bacteria, instead of methionine, there is a small corrected methionine called by what name? By what name? 4-mile methionine. It's a small correction. Understood? Understood?
Yes,
Yes,
sir.
sir.
So this is 4-mile methionine. This extra group is called 4-mile. Understood?
Yes, sir.
to that COOH group instead if there is a hydrogen, that is methionine. Okay, removing the hydrogen, if this C double bond OH, that is the aldehyde group comes, mean what is it? What is it? What is the name?
Methionine.
No. If you understood my point to this structure, okay, without having this, instead, if you have a hydrogen, if you have a, what is it? Hydrogen, what is this? It is methionine. Clear?
Yes, sir.
Yes, sir.
This is what we call methionine. Instead of that hydrogen, if this group is present, what is the name of this group? Formyl group. Now, what is the name of this amino acid?
Formyl methionine.
Formyl methionine. Can you understand the difference between methionine and formyl methionine?
Yes, sir.
Yes, sir.
Yes. But this structure knowledge is not necessary for you. Understood? This structure knowledge is not necessary for you. But then you might ask, then why are you telling, sir? The reason, if I just tell formyl methionine, okay, what will stay in your mind? Tell me. Just there is formyl methionine mean, what is the meaning? No, you have to understand it. No need to memorize it. Clear?
Yes, sir.
Yes,
I am not asking you to draw this structure or practice this structure or, okay, in the exam paper, draw this structure. That is not my expectation. But a student, if a sir telling formyl methionine, the exam paper formyl methionine mean, this explanation is the one going to improve your memory. Right? So, both of them are nitrogen containing, sulfur containing amino acids. What is in the side chain?
Sulfur.
Sulfur. So methionine is a sulfur containing amino acid. Formyl methionine is also
Sulfur containing amino acid.
sulfur containing amino acid. Both of them are initiate amino acids. Why? They will come in the front of the chain. Clear?
Yes, sir.
Right. So, some instances, that mean who have formyl methionine? Who have formyl methionine? Domain bacteria. Other two are having
Methionine.
methionine. Understood?
Yes, sir.
Good. Another, now that protein synthesis story, are you clear?
Yes, sir.
Yes. Now coming to another character to compare response to antibiotics. What are the antibiotics? Read the name of the antibiotics.
Streptomycin,
Streptomycin, chloramphenicol. What are the antibiotics?
Streptomycin and chloramphenicol.
Chloramphenicol and streptomycin. When you add streptomycin or chloramphenicol to a petri dish and introduce some domain bacteria member in that petri dish, in that medium, that bacteria will not grow. Understood? Did you understand what I told?
Yes, sir.
Okay. Now, in laboratories, there will be bacterial culture. Can you see bacterial culture? So these are, what is it? Bacterial culture. Bacteria grown in a petri dish. Okay, before introducing the bacteria, we will be preparing a medium. It will be a jelly-like medium, what we call, okay, culture medium we say. Now, in this picture, can you see the bacteria has grown?
Yes, sir.
Yes.
Yes, sir.
In this medium, if you have mixed some amount of streptomycin or chlorophenicol, bacteria will not grow. Understood?
Yes, sir.
But if you introduce some archaea bacteria, it will grow. Its growth is not inhibited by
Antibiotics.
not all the antibiotics.
Okay, sir.
Okay, not all the antibiotics. Okay, what are the antibiotics?
Streptomycin and chloramphenicol.
Streptomycin and chlorophenicol. Limit your idea to those two. Don't generalize it. Generalization will not help you. All the, okay, now say growth of archaea bacteria is inhibited by antibiotics. Mean one side, not inhibited by antibiotics mean growth of archaea bacteria is not inhibited by antibiotics mean one side it is correct because chlorophenicol doesn't inhibit. Correct?
Yes, sir.
Growth of archaea bacteria is inhibited by any antibiotic mean
It's wrong.
it's wrong. Understood?
Yes, sir.
Yes, sir.
So their growth is inhibited by antibiotics. Now say a person take, now actually we give chlorophenicol as an antibiotic to some diseases also. Understood?
Yes, sir.
Some diseases also. In animals we give, what is it? Chlorophenicol, streptomycin. I have given chlorophenicol to chick birds. I was growing broiler chicks after my A levels before entering the university. Before entering the university, I was growing broiler chick. That broiler chicken you buy. Okay.
Yes, sir.
So there are reasons for this. For an example, now if we take streptomycin, the streptomycin is important. So importantly, what it does is they are destroying the peptidoglycan, or they are stopping the peptidoglycan synthesis in the bacteria. That's how bacteria growth is stopped by the streptomycin. Understood? Understood my idea?
Yes, sir.
Now in the archaea bacteria, is there peptidoglycan?
No, sir.
No. So if you give an agent that inhibits peptidoglycan synthesis, will it stop the growth of archaebacteria?
No, sir.
No,
No,
sir.
sir.
This is what's happening. That's why archaebacteria is growing without considering whether streptomycin, they have chlorophenicol there. Understood?
Yes, sir.
Right. So, hope you all understood the response to antibiotics, streptomycin and chlorophenicol. Are you clear?
Clear, sir.
Thereafter, another comparison given. Growth at temperature greater than how much?
100 degrees Celsius.
100 degrees
100
Celsius.
degrees. That means they should be high temperature tolerant. Correct?
Yes, sir.
High temperature tolerance is not there for whom?
Domain bacteria.
Domain bacteria or
Domain
Eukarya.
eukarya.
domain eukarya. It is there for some species of
Domain archaea.
domain archaea. Those species are normally called by the name, what is it?
thermophils.
Thermophiles. They are heat loving organisms. They love to live in high temperature, hot water springs, volcanic pits like places are their choices of life. Understood?
Yes, sir.
So it is also not told all of them. One category of
Domain archaea.
domain archaea only thermophiles. There are so many different files there in the domain archaea. All of them are together called by the name extremophiles. What is the name given?
Extremophiles.
Extremophiles. So if you take extremophiles, okay, there are acidophiles there. They love to live in which environment? Low
Acidic.
pH. Alkaliphiles are there. They love to live in
Basic environments.
basic environment above 9 pH. Anaerobes, they are also coming under extremophiles. Understood? Halophiles are there. Like this, cryophiles are there. Osmophiles are there. Okay? Radio resistance are there. Xerophiles are there. Like that, many extremophiles are there. All of them are not in the syllabus. You have only thermophiles and halophiles in the syllabus. Understood? And anaerobes in the syllabus. What are the three extremophiles in the syllabus?
Thermophiles, halophiles, and
Thermophiles, halophiles, and
Anaerobes.
anaerobes. Those three are in syllabus. Others are not in syllabus. So could you please complete the seventh one?
Done, sir.
Right. And finally, their habitats. Okay, normal habitats are choices of domain bacteria and
Domain eukarya.
okay. But archaebacteria, they prefer to live in
Extreme environment.
extreme environments. Thing is, they are occupying, what is it? That is actually a kind of a successing formula. Understood? It's a kind of succeeding when no one is ready to occupy some resource, correct? If you have a strategy to occupy it, you are the winner. Clear? They will not even think of coming near to that. So, the habitats, extreme environmental conditions are one of their major choices, but you know, they can live in moderate environments also, correct?
Yes, sir.
Yes, sir.
Okay, they can live in moderate environments also, though it is mentioned here, extreme environment. The same resource book is the one telling in another place, they are possible to live in
Moderate environment.
moderate
Moderate.
environment. Understood?
Yes, sir.
Right. So complete the habitats.
Done, sir.
Okay. Thereafter, finally, example. Here, all these are scientific names written. What are the scientific name written? Nostoc. Read the second one.
Anabina.
Anabina. Third one. Escherichia coli. Escherichia coli. And last one,
Salmonella typhi.
Salmonella typhi. Among these, two are cyanobacteria, two are bacteria. Okay. Bacteria, Escherichia coli, Salmonella typhi. Cyanobacteria, whom and whom? Nostoc and Anabina. So when you are writing example, write the bacterial example under bacteria, write the cyanobacterial example under cyanobacteria. Understood?
Yes, sir.
Complete that one, keep the archaebacteria. After you finish the bacterial example, tell me. Hi. Came. Completed?
Yes, sir.
Yes.
Right. Archaebacteria, what are the examples given to you? Read the archaebacteria examples. Read it.
Methanococcus,
Methanococcus,
Thermococcus,
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 Halobacterium,
and Halobacterium. Methanococcus is an anaerobic bacteria. Who is Methanococcus?
and anaerobic.
It's an anaerobe. Anaerobes are extremophiles, no? Correct?
Yes, sir.
So under Methanococcus, you have to mark, what is it? It is an anaerobe or anaerobe. Thermococcus is, name tells you, what is it? It is a
Thermophile,
thermophile. And Halobacterium is a
halophile.
halophile. Salt environment, highly saline environment, Halobacterium thrives. Clear?
Yes, sir.
Good. Complete it. Under each of the example in the brackets, put what kind of organisms they are.
Done, sir.
Right. So with this one, the comparison is over. With this one, the comparison is over. Next heading we are coming to is kingdom. What is it?
Protista.
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 You know that one. Okay. I was shouting on this kingdom Protista from the beginning of the lesson. What I was telling is, when you complete kingdom Protista, my children, you are ready for the first term. Okay, you are ready for the first term. So in your school, when you are sitting first term examination, now your school would have now started again, no? Correct? Again, it will be going with nucleic acid, carbohydrate, yes or no?
Yeah, sir.
Yes, sir.
So they will be coming to an end, mostly. Okay, now February mean March, April, May, June. Okay, to my knowledge, not in March, maybe June, your first term examination. Understood?
Yes, sir.
Yes, sir.
Thing it is, in the first term examination, you all have to take very good score. That is my task. I am giving all these works with only that hope. Understood?
Yes, sir.
And hope is that your first exam should be, what is it? Super performed. Then you will come to a confident and okay, you will start to love your performance. That will push you to the second term. So that's why after giving these essays, I'm continuously waiting. Who have completed all the essays? Amna, Faisal, Amira, Tariq. You two have completed all the essays I have given, correct?
Yes, sir.
Now
Yes.
new people are there. Now Afla Rafaidin is there. She is new. I have not given her. Nada is new. Okay, Zainab Zanir is new. But Rima is not new. Humaira is not new. Avasha is not new. Okay. So if you complete it only, anyway, I'm going to start. I can't wait. I'm going to start my other works. இனி போட போறேன் வேலைகளை தொடர்ந்து அடுத்ததா வேலைகள் போட போறேன். இதை நீங்க கம்ப்ளீட் பண்ணணும்னா நான் பார்த்து கொண்டிருந்தேன். பட் the expected speed is not there. The expected speed is not there. Okay. So inshallah today onwards my other works will be coming. repeatedly, those who have completed the essays, okay, for every 3 days, write the same essay, one choosing from the beginning, number one, repeat it for every, what is it, 3 days, you write one essay again without seeing the previous scheme or previous writing. Understood?
Yes.
Yes.
Okay, I have plenty of works for you.
Sir,
Yes,
sir, practicing only the essays that you have given for first term is enough?
95% enough.
Okay, sir.
For the moment, enough. 1 or 2 essays can go above that. Okay. But
Okay,
first
sir.
term, no. First term, oh, now one more essay there. That is this time an essay came in the 2022 paper. What is the essay? Have you gone through the 2022 paper? Anyone?
Yes, sir.
In the essay? First essay. An essay on meiosis. Correct?
Yes, sir.
Yeah, that essay. Some greater chances there that essay to come in the, what is it? Your first term paper. Understood? Recently came one, no? As it is a recent one, there's a bigger chance for, what is it? That essay to come. Those types of things happen. Anyway, before your first term examination, official first term examination in school, we have to keep some first term examinations and train you. Understood?
Yes, sir.
So if I complete Protista, I will fix the first term examination. Okay, at the end of this February. At the end of this February, I will fix my first term examination with you. Clear?
Yes, sir.
Your papers are in the ready form. Schemes are in the ready form. I have less things to do on that. I am in the ready form. So, get ready with it. Don't delay the works. Okay. Today, probably the next work will be starting. I gave you cellular basis of life structure papers now. How my children?
Yes, sir.
Did I give you? Avasha?
Yes, sir.
How many papers I gave? 3 papers?
Yes, sir.
Sir.
3 papers. Cellular basis of life MCQ paper?
No, sir.
No,
No, sir.
I didn't give you.
No, sir.
So those papers are waiting for you. Any order when done. Potista vanditale refunds or order done. Works done. Okay. But do it. Okay. You know, I will time the things. I will time the things. Understood? Now, essays I gave you, just I gave you. Okay. Different people are doing at different time. But MCQ paper, structure paper, that cannot be done, Putha. Understood? One child does it today, another child does it after 10 days mean. Will there be a regularity? How can I give the scheme? So get ready for the cell biology MCQ paper. How many days you want? How many days you want to get ready for the cell biology MCQ paper? One essay question, Zainab, 30 minutes. That is the maximum time. Whether the essay finishes or not, we don't care. 30 minutes. Can't give more than 30 minutes. In exam papers, keep in the mind, structure paper, essay paper, both will be given to you at once. You have to do the essay first. Finish the essay, then go to the what?
Structured essay.
Structure paper. The structure paper you will get, you will go on writing. Sometimes you will be eating the time of
Essay questions.
essay question also. That commonly happens. So the recommended is, I can only recommend. Now, if we are choosing the other track, I can't force you. Understood?
Yes, sir.
So you have to get ready for it. That is very, very important. Right. Get ready. So I'm asking this question at you. How long time you want to complete? So to do the cell biology examination, how to get prepared for the cell biology examination. Cell biology mean only cell biology, not cell division.
2 days.
You decide. I will keep my speaker and microphone in mute. You discuss and tell me. You can discuss among you, that I will not hear. You discuss. Have you discussed? Have you discussed? Hello? Hello, can you hear me?
Yes, sir.
Have you discussed when to do the cell biology paper? I didn't hear anything. because I have switched off the audio. Allowed you to discuss. What about you? Did you make a decision?
So is it just a MCQ paper?
Yeah, 50 MCQ.
Sir,
Hmm.
I got disconnected due to power cut. Can you please repeat?
A cell biology MCQ paper. Ready to do. When are you going to do? How much time you want to get prepared?
So on Sunday?
Anything is okay. I will give you the time. No problem. You have enough time. But all of you decide a time and tell me. சரியா செய்யணும். டைமுக்கு செய்யணும். That is very important.
So the whole unit.
Not whole unit, Baba, cell biology.
Okay,
Not
sir.
chemical basis, not cell division, not metabolism and enzyme, not photosynthesis, not respiration. Cell biology from Robert Hooke to extracellular matrix. Tell me, Sunday okay, everyone?
Okay, sir.
Is Sunday okay with everyone?
The timing?
You are the one going to decide it. But anyway, I will be flexible in that. I can't fix a time for that. Different people have different classes. Okay. Saturday night, I will upload the paper. Okay. Before Sunday night, 10 p.m., you do and send the answers. Okay, no?
Okay, sir.
Okay, sir.
I'm being flexible with you now. I don't know your other class timetables, those things I'm not much aware of it. And synchronizing all the children to one time is very difficult. Okay, so I'm being flexible in it. Okay, Saturday night I will send the paper before, what is it? Sunday night, 10 p.m. you send me the answers. Understood?
Yes, sir.
Right. So you have to continuously get ready. அடுத்த எக்ஸாம் சொல்லிக்கிட்டு இருப்பேன். அதுக்கு ரெடி ஆகுங்க, அவ்வளவுதான் சொல்லலாம். பொட்டிஸ்டா முடியுதுங்கிறது, பொட்டிஸ்டாக்கு வந்துட்டோங்கிறது, பொட்டிஸ்டா you are at பொட்டிஸ்டா mean what is it? The powerful practices are starting. They wait for the practices. First, let's complete the unit number 2. Then we will go to the practices on unit number 3. After you are becoming very thorough in the unit number 2, we will go to the unit number 3. Unless you are becoming thorough in the unit number 2, I will not get into that. Avasha, ready to do the exam on Sunday?
Yes, sir.
Okay. So, inshallah, we will meet. Protista we will discuss on the coming class. You get ready for the works. Sunday, okay, Saturday night, I will place the exam paper to you. Okay?
Okay, sir.
Okay. Bye then. Assalamu alaikum.
Walaikum
Jazakallah,
assalam.
Barakallah.